OmicsOffice ExpressionExplorer allows visualization of expression data in an unprecedented depth by comprehensively characterizing both expression and differential expression results. This tool allows the data to be displayed in such a manner that makes it easy to discover relevant facts about the underlying biology. In this particular example, we will be using Affymetrix microarray data to display different functionalities of ExpressionExplorer. We will demonstrate how we can load data files in text format where signal intensities have been normalized, as well as data that has undergone differential expression analysis between two groups using a third-party software. Once the comparison file displaying differential expression analysis has been loaded, we can go on to define the different comparison groups. In this case, fold change, p-value, and adjusted p-value. We can also specify different annotations that we want our data to be integrated with. It is also recommended to configure the relevant settings for our analysis purposes, such as adding relevant annotation sources, defining the k-means clustering settings, as well as loading any custom target lists for comparisons with the experimental dataset. We can save these configuration settings to be run at any time. Once the analysis has been run, ExpressionExplorer will present us with various tabs that refer to specific visualizations. The overview page shows general information on the loaded data and main results and answers questions such as which comparisons were analyzed, globally, how many significant or non-significant differences were found in each comparison, which are the significant genes in each comparison. The navigation panel on the left-hand side gives an overview of the information on data import, input files, and experimental design. This differential expression overview bar chart and table shows the number of targets in differential expression groups for each of the imported comparison. The visualizations on sample expression similarity page addresses the correlation and similarity between samples. OmicsOffice capitalizes on the Spotfire capabilities where the tables and visualizations are synchronized and are altered in real time. Apart from the correlation plots, the data is also presented using principal component analysis to allow the user to look at the variations in their data and to identify global outliers or outliers within defined experimental groups. The visualizations on sample expression box plots page tries to identify the global distribution of expression values across the samples and between groups with the aim of determining whether the samples have been normalized well. Top expressed genes page tries to identify the targets that show the highest expression in experimental groups, as well as how these top expressed targets behave across groups. The top number needed can be specified according to user needs. As mentioned previously, all these tables and graphs can be changed in real time. Additionally, the x-axis can be adjusted if we wanted to alter the depth of the analysis, in this case samples versus groups. The expression ranks page tries to identify the targets that are to be found in the upper, lower, median ranks of expression in any given group, as well as the expression ranks that may have been allocated to a specific gene within the groups. It also informs us how the expression ranks behave across groups and which targets show large rank differences. The rank target expression table shows the expression ranks for selected experimental groups as well as the average ranks. Select lines to show corresponding targets in the bar chart. Differential expression volcano page represents data so as to identify which targets are significantly up or down regulated in any given comparison. The page also represents how these sets of targets behave in different comparisons and the intersection between data derived from experiment-derived and custom lists. Targets expressed higher in the target group are on the right, whilst those expressed higher in the control group are on the left. Fold change of 1 and -1 mean twice as much or half as much expression in target compared with control group. Furthermore, intersection of lists can be used to mark the gene lists on a specific volcano plot. However, if you're interested in looking at the impact of these gene lists across other comparisons, then you can select volcano plots for as many comparisons as you like, and these gene lists will be marked on them. You can also mark targets using specific genes or annotations. Differential expression comparison view uses a qualitative scale to determine how different sets of targets behave in different comparisons. Additionally, this comparison also assesses the intersection between experiment-derived data and custom lists and how they are differentially expressed across comparisons. The information is further enhanced by the line chart that shows the expression values of selected targets across selected groups. If more than one target has been selected, multiple lines will show in the line chart. Visualizations on the list functional characterization page try to answer what functions can be assigned to lists of significant up or down regulated, top expressed lists, or k-means clusters based on known biological annotations, for example, gene ontology, etc. This visualization shows the number of enriched terms in each annotation category used in enrichment analysis at the specified FDR threshold for selected lists. Select any bar segments to show details on enriched terms in the tree chart and table. The list similarity heat map shows the similarity of functional terms assigned to each list using Jacquard similarity, that is, the proportion of shared terms in the union of all terms from both lists. Red refers to high similarity, blue refers to no similarity. Visualizations on the lists overlap page tries to answer whether there are any lists that share similarities with known target lists from previous experiments or public databases. Please note that this page is only available if custom list import has been configured in analysis profile settings. The overlap between lists in analysis and with custom target lists is presented in the overlap table. Overlaps are shown as a proportion of shared targets in either of the lists. That is, an overlap of 0.5 means that 50% of the list is shared with the other list. The premise being that shared targets might indicate similarity in biological significance of lists. You are easily able to select the list that you would like to see the overlaps for. For further information, please go to www.integromics.com